pleasure, and we're lucky to have Dr. Jessica Hoffman here from UAB, University of Alabama, Birmingham. Jessica is an expert in comparative models of aging and longevity, so studying lots of species to really try and understand how and why they live so long, or even closely related species have such drastically different lifespans. Dr. Hoffman spent a lot of time in the Southeast, it seems, doing her undergrad at Clemson, her PhD at Georgia with Daniel Promislau. She moved with him to the University of Washington, where she and I overlapped. Well, we overlapped later on. And then she moved on to start a postdoc at UAB with Steve Ostad. Just a year and a half ago, she was awarded a prestigious K99 R00 Kangaroo Fellowship. That transitioned from postdoc to a faculty position through the National Institute on Aging, where she's trying to study the metabolomics of mice, dogs, and fruit flies to really try and understand what determines, or what are some of the mechanisms underlying the differences in lifespans across these species. And today, I think she'll tell us a little bit about that. So I hope you'll all join me in welcoming Dr. Hoffman, and we look forward to your talk.
Okay. Great, thanks, Noah. So like you said, today I'm going to talk about comparative metabolomics. I'm going to tell you two stories that we've kind of been working on in the lab and kind of coming to different conclusions about aging across species. So in case some of y'all aren't aging biologists, which I assume not, it might not be surprising to you to know that life expectancies have been increasing dramatically over the last 100 years. So this is just some data from the United States since about the 1930s, looking at life expectancy at birth. So this is the number of years an individual can expect to live from the point at which they're born. And you can see that both for males in blue and females in red. This is going to be throughout the talk. You'll see males in blue, females in red. You can see a significant increase in life expectancy, where now most females, when they're born, can expect to live to be about 80 years old. Life expectancy at 65, or the number of years you can expect to live if you make it to 65, is also going up at this same rate. And now it's up to almost 88 years for a female. To where if you live to be 65 years old, you can expect to live to 88 years old here in the United States. So there's been these dramatic increases in lifespan, mostly due to our developments in public health, clean water, vaccines, all these different public health initiatives that have really helped us with our longevity and aging. However, there's not necessarily been an increase in health span. So health span kind of refers to this amount of healthy life an organism or human lives. So you can imagine you're chugging along, you reach 65, you develop some sort of disease, and you die at 70. Well, now we're living longer, but unfortunately, all we've actually done for the most part is increase this period of morbidity. We haven't really increased the healthy lifespan of most individuals. So why is this? Why do we age in general? And why are we developing all these age-related diseases, even though we seem to be having improvements in medical intervention? And as biologists, we go back to the model organisms usually. So yeast, worms, flies, mice, the 4 standard laboratory organisms. We can manipulate genes, the environment, look at their interactions to begin to understand why individuals, different organisms age. And just as evolutionary biologists, we kind of want to think about why aging maybe was selected for, right? So we don't just live, reproduce, die, right? We age. Why do we age? Why have we not been selected to just reproduce and die? And one of the main theories is antagonistic pleiotropy, which is kind of depicted here, where you can imagine you have some gene that in the wild increases your reproductive success, but then decreases longevity and health. And so maybe later on in life, this gene is actually bad for you, but because you've already reproduced, it's not selected for. Conversely, what we found with these model organisms is many, many different genes that we can mutate, and they actually live longer and they live healthier. So acting on these genes not only increases lifespan, but also health span. However, there is always some reproductive or fitness consequence to these genes, which is why they aren't selected for. So These laboratory models are great. However, they don't really recapitulate all the phenotypes seen in the wild. They've been wildly selected for very strange phenotypes at times. If you talk with Steve Osted, he will tell you a lab mouse is its own species almost. It's not like your standard wild type mice. So one of the ways that we think about maybe getting around that is doing comparative analyses. So looking both across and within species to determine conserved mechanisms of aging. And some of these patterns that have been known for a while, there's a correlation between body mass and longevity. Both in blue, these are things that fly, and red, things that are terrestrial. And you can see there's a strong positive correlation in both groups. However, there are outliers everywhere. And those outliers can also give us some interesting information on why they are aging either worse or aging better. A similar one is that we know that there's a negative correlation between basal metabolic rate and maximum lifespan, or just lifespan. So animals that are larger have lower basal metabolic rates. So these are patterns that we probably would not have picked up on if it weren't we're looking across species, that then give us some questions that we can begin to look at within species. So as evolutionary biologists, aging biologists, just biologists in general, we're often concerned with connecting genotype to phenotype. So these genes are what are actually selected on. These are what evolution can work on. However, it's the phenotype that is selected for, right? However, how do we get from genes to a specific phenotype, or in our case, aging and longevity? Part of the problem is that there's so many levels between the genotype and phenotype. It's not just this gene gives us this phenotype. Genes produce transcripts, which produce proteins, which interact with the microbiome, the metabolome, the lipidome, which then themselves produce the phenotype. These all interact with each other within a level and across levels. And on top of that, the environment affects every single level. And so that's its own issue you've got to deal with, as we know the environment affects specific phenotypes. So because of this, I've chosen to focus kind of on this level right here, the microbiome, or not the microbiome, the metabolome sitting with, because it actually allows us to integrate the genome and the environment. So the metabolomics in itself is just the systematic study of all the metabolites and their functional roles. And then by integrating the genome in the environment, I mean that there are metabolites that come from the environment themselves that we pick up. So if any of you are having your lunchtime pick me up with your caffeine, we pick up that caffeine in our metabolomic screens. Similarly, metabolites that are made endogenously in the body, something like lactic acid, also are picked up on our screen, as are those that come from potentially our diet or an environment, as well as are made within our bodies. So the metabolome really integrates the genome in the environment in a way that other levels of organization don't. And so that's why I've chosen to really look at this to see if we can find metabolites and metabolic pathways that are associated with aging and longevity. Can we then really begin to understand why we're aging? So this is kind of the global metabolic pathway for those of you who are interested, the KEGG pathway. This slide's actually a little out of date, but every black dot is a metabolite that we can pick up on our global metabolomic screens. So this is work I've been doing with Dean Jones. And so you can see the majority of the metabolome can be picked up with our mass spec techniques. However, there are certain groups like the glycans that do not get picked up. So anything you see today is not going to include any glycans. Lipids, we can change settings to do lipidomics, which I've actually done a little bit with my dogs, but I'm not going to talk about that today. And one thing I want to point out that's really interesting is we do pick up the xenobiotics. So these are all the foreign metabolites. So these are things that are from the outside in your body, and we can pick the majority up, as well as the metabolites that they're broken down to within our bodies. So I'm going to tell you kind of two stories today, and I'm going to start with a comparative metabolomics of the companion dog. So many of you might have your pet dogs next to you. Mine is right behind me at the moment, since we're all working from home. And you can see there is great phenotypic variability in the dog. There are dogs with this is the bulldog. This is UGA's mascot. So short, squatty, overweight, not very healthy breeds. We get the Australian Shepherd, high energy herding breeds. we get lazy breeds. So we have selected these dogs for a range of morphological variation. So in essence, they become an evolution experiment themselves that we're trying to harness to really understand what's going on with the dogs. What might be most interesting though is their size variation. So there's extreme size variation in the dog. This is an Irish wolfhound. It can expect to only live about 8 or so years, and these tiny little dogs can expect to live 15 to 16 years. So this huge size variation is actually associated with longevity. So this is opposite what we saw across species, where across species, the bigger individuals are longer lived. The larger, the elephants, the whales, longer lived than the small mice. However, within a species, it seems that the smaller individuals are long lived. So this is just some data from about 5,000 dogs that died in the UK that I got from a collaborator. And you can see both in mixed breeds, which live longer than purebreds, there's a negative correlation between size and longevity. So why is this? So for years, it's been assumed that this was due to changes in the growth hormone IGF-1 axis due to results that have been seen in mice. So this is kind of the IGF-1 axis. Your hypothalamus produces GHRH, which activates the pituitary, which then activates growth hormone in the liver to produce IGF-1. And it's thought that higher levels of IGF-1 lead to increased size and decreased longevity. And this is backed up with, like I said, with data in mice. So we know that growth hormone mutations increase lifespan in mice. So this is data from growth hormone releasing hormone knockout mice. So these guys up here. And you can see in both males and females that those that are knockouts, so they have lower levels of growth hormone, live longer. These are some pictures actually of Amesdorff and some other growth hormone mutant mice, but you can see they're little tiny cute mice. So they're very small, but they're long-lived. So then it was also thought that, oh, sorry, I should say this first. So dogs also seem to have lower growth hormone in smaller dogs. It's not been well studied. Growth hormone is very hard to measure, so everyone tends to use IGF-1 as a proxy. But there's been one study that measured growth hormone concentrations in beagles here at the bottom, so smaller dogs, and Great Danes here at the top. And you can see the bigger Great Danes have significantly higher levels of growth hormone. But this has not been done in many breeds. Like I said, it's really hard to measure growth hormone. And the collaborator we wanted even to use to work with growth hormone can't even do the assay anymore because the person that did it retired and they had no one take over. So it's just a really difficult assay to do in dogs. So people tend to then use IGF-1. So if you look here, remember growth hormone activates IGF-1, and this is what people really think is probably driving it all. So, however, the results in mice are a little iffy. So it's been shown early on that IGF-1 knockdown, these are heterozygous because if you knock out IGF-1, it's embryonic lethal, but are longer lived in males and females. However, this study was repeated in a different genetic background, and it looks like IGF-1 might not actually increase lifespan. So there's a lot of debate on if IGF-1 is really this thing that's driving this increased lifespan in these mice, because growth hormone reduction obviously increases lifespan. It's been shown in over a half a dozen models that works. IGF reduction, maybe not so much, but this is what people who work on dogs thinks is driving the effects. So this is just some data in males. Females show the same thing, that if you correlate weight with IGF-1 levels, there's a significant positive correlation. So it's been assumed this higher IGF-1 leads to increased size and leads to decreased longevity. However, because of the iffiness in the mice, and there's a lot of nuances where Rottweilers, big dogs, actually carry an allele that small dogs have for IGF-1. So you would think they'd be small and long-lived, but they're big and short-lived. So I'm not fully convinced that this is what's driving this. So because of this, we wanted to do an experiment to see maybe if something else is going on. That can we find some novel hypotheses on why small dogs live longer? So this is a study where we took blood from 112 dogs across 3 different locations. This should be Missouri. Sorry, that's a typo right there. So Birmingham, here I am, the Columbia, Missouri Veterinary School, and then in San Antonio, Texas at a spay neuter clinic. So all the dogs were at least 1 year of age, so they're fully grown. And then we did global metabolomics, and then we did do lipidomics, but I'm not going to talk about that specifically. So we did these global metabolomics, and we want to know what is associated with longevity, or in this case, weight, right? Because we're using weight as a proxy for longevity. So we took all our metabolomic samples, we did our PCA, and the first thing we actually found out is that sampling location made a much bigger difference than what we expected. I threw this in here not because it has anything to do with our results, but if any of you are doing any sort of omics, especially metabolomics where the environment matters, and you're sampling from different locations, make sure you control for your location effect before you do any analyses, or your data will get kind of messed up. So even though these were some of the same EDTA tubes, the same veterinarian between Birmingham and San Antonio drew the samples, and so the sample handling was all the same, yet we still see this huge location effect that had to be pulled out before we could actually look for those metabolites that are associated with weight in the dog. So what did we find? We ended up looking at about 3,000 metabolites. What types of metabolites did we find that were associated with body weight? The number one pathway that came out was the tryptophan degradation pathway. And I don't know if y'all know anything about this. I didn't know much about this until recently. So most of us think about tryptophan as an amino acid that gets converted into serotonin. It's where the whole, you eat the turkey at Thanksgiving, you get full, and serotonin becomes melatonin, you get sleepy. So tryptophan to serotonin is always kind of what people think of. However, actually 95% of tryptophan is actually broken down into kynurenine. It's not shifted towards serotonin, it's broken down to kynurenine. And so this specific pathway is what we found that seemed to be different between the large and small dogs. And so if we look at the data, so these are 6 different metabolites that we measured in the dog that were in the tryptophan pathway. Here's a schematic kind of quickly of the mammalian version of tryptophan degradation. It doesn't have all these metabolites on it because there are more intermediate steps. And what you can see that is that the small dogs have higher levels of all of these tryptophan metabolites, suggesting that there seems to be some sort of upregulation in this pathway in the small dogs. And so then the question becomes, maybe this is something that might be driving this small size, longer life phenotype that we're seeing. We should mention that both tryptophan and kynurenine, those two that matter the most that I mentioned, don't actually show a significant negative association, but it's trending, but it's not significant. But again, none of the metabolites in this pathway go up with weight. They either have no statistical difference or they went down, where the larger dogs have lower levels. So this was interesting. And so we were thinking, well, is this something that actually happens in other species? And actually, a year before, I'd done a study with the Southwest Primate Research Center on marmosets. And we actually showed that low tryptophan was predictive of mortality. So we took blood from about 100 different marmosets, and then we followed them for 2 years and asked who died within those 2 years. So here are the actual values, and you can see that for those animals that died within our 2-year follow-up, they had lower levels of tryptophan. And this is after controlling for age and weight. So it's not just that the older animals had lower levels and so they were more likely to die. It actually is, it seems to be that low tryptophan might be predictive of mortality in these animals as well. So now we've got 2 species where something appears to be going on within the tryptophan metabolism pathway. So we, of course, wanted to go back to the laboratory and see, is this actually causative? Because you can imagine that a lot of stuff that we find that's associated with aging and longevity in species is actually just a byproduct and not actually causative of anything related to aging. So we went back into the laboratory to use the fruit fly to ask, can we manipulate this pathway and increase lifespan or health of the flies? And so the next couple of slides, I'm going to show you some of this is data I got literally yesterday, so I don't promise. It's all very preliminary. The graphs aren't pretty, but these are two really talented undergraduate students who have done the majority of this work on the fly. So the first thing we did was the easiest experiment there can be. Add tryptophan to the food and see what happens. So we used 3 different doses based on previously published papers, but no one had actually followed it out for longevity. So we had a low dose, a medium dose, and a high dose. And what you can see is that the high dose kills the flies immediately. So a lot of tryptophan in adulthood in their diet, really bad. The medium and low dose seems to have no real effect. I'm only showing you one genetic background here. We did this in 3 genetic backgrounds, and in every background, the high dose is extremely lethal. The medium and low doses sometimes are the same as the control, oftentimes actually also slightly more lethal, increasing mortality. So it seems to be adding tryptophan into the diet, at least in flies, not a good thing. So some of the other previous studies had actually done this in development. So they added it into the food during development, and then they were measuring something else, and they were asking about a specific neurodegenerative phenotype in young flies. So we said, okay, let's add it to the food for the larvae, and if the larvae eat it, but then take it away as adults. And here we begin to see a little bit more of a maybe an effect. So this is one of the genotypes. This is the genotype that showed the probably best effect. The other 2 were more closer to the controls. And in the controls, in the solid lines here, again, males are blue, females red. You can see at the low dose, there seems to be actually an increase in lifespan, at least in median lifespan in the females, nothing in maximum. It does not seem like maximum is affected at all with tryptophan manipulation. The medium dose seems to increase in both sexes. And then in the high dose, again, it looks like there might be a female bias. The one thing I'll say that we need to redo this experiment, because you can see, especially in the control females, they had a really sharp mortality right around 45 days. And if we were to remove this, it would be shifted further out. So I'm not sure I 100% believe this, but at least it's a little more promising than adding it as an adult. Because I really did not expect this, especially this high dose that people feed to flies fairly often, that it would actually, if you take them out past 7 days, it turns out it's not good for them. Okay, so what about a genetic manipulation? So flies have a slightly less complicated tryptophan pathway. And because fly people are weird, I don't know if there's any other Drosophila people here, they like to name their genes after the phenotype of the mutant. So the wild type actually will have a different phenotype than the name. So this vermilion is the same thing, mammalian, go way back. TDO, IDO, these are the same. I should have added that in, I apologize. But the Drosophila people just name it differently. So we were interested in looking at this mutation, this V vermilion mutation, which then is going to increase tryptophan genetically. And it's also going to block this reaction. We are hoping to add in this mutation as well, this CEMinar. However, the flies all died in my hands, so I've got to reorder them, and we'll see if hopefully, and them all dying might be an indication that they're not a very fit mutation at all. So this is our longevity curves that we just finished. And so the V's are in the solid and the dashed, and then the dotted is our control. And you can see that there actually appears to be an increase in lifespan in these flies, and at least in this one trial. Again, it needs to be repeated. These are all super preliminary, but this looks more promising to knock down or to increase tryptophan genetically throughout the life versus adding it as a supplement into the diet. And then again, as evolutionary biologists, we're also interested in what is the effect on fitness? How are these flies just as fit? Why is this mutation not been selected for? So an undergrad in the lab just finished several fitness assays. The sample sizes are not great. So again, it's very preliminary. The first one she did is called a larval crawling assay. So you put a bunch of larvae in this middle circle, you let them go for 10 minutes, they're gonna search for food, and then you count how many have actually moved. So it's a measure of larval fitness and their ability to move to find food. And so unfortunately with this genotype, they develop a little slower and she didn't get any, but you can see there's no real difference. They all moved. These are all pretty fit larvae in the mutation. This is another control that we added in that unfortunately wasn't in our longevity experiment. Then we also looked at just reproduction. So we took one male and one female, let them mate for 48 hours, lay eggs, and then count how many offspring are produced. And here we're getting a little more variation, but it seems to be a little all over the place, so I'm not exactly sure what this means. So here's the mutation we care about, and it looks like they're not producing nearly as many offspring. However, this fly also has the mutation we care about, and they are. And this one, again, does not have the mutation we want, and they're low. So the sample size isn't great. I'm not sure exactly what this means, and we're hoping to add in some RNAi, some other ways to knock down these genes that maybe will give us a little more of an indication of what actually is going on. She also did some more health assays, which are kind of later fitness. One is climbing ability, where you knock down the flies, you give them 10 seconds to climb up, and then you see how many have passed this 5-centimeter line within those 10 seconds. And you can see in both females and in males, there's no difference in these guys. I will say though, these are very young flies. They're only 7 days old. This might change with age, and that's something we're planning to do later on. And the last thing she did with these flies was do a heat stress assay. So this is where you put these flies at 38 degrees Celsius and you just wait for them to die. And so they all die within 7 hours, some much, much sooner. And again, these lines are a little thin, so I apologize. The solid lines are the one we care about the most. And you can see they might be a little longer lived, but again, this other mutation with the V in it is short lived. So it's nothing super exciting yet. It's maybe a little more promising than the supplement, but there's still a lot of work to do. I wanted to throw it in though, because this is data, like I said, I just got. And as all of you know, we've been at home for so long, it was nice to get kind of new data for the first time. So kind of part one conclusions is that we find that, I just want to add in that location has significant effect on the impacts of metabolomic variation. So this is something to think about if you're going to be doing any metabolomic sampling across locations. Is it the microbiome? Is it other environmental factors? Is it actually some sort of sample handling that we didn't realize? There are many, many different things that I think could be driving this, but it's just an issue to think about when you're running these types of larger across distance experiments. So we found that tryptophan appears to be differently regulated between large and small dogs, and that low tryptophan predicts mortality in marmosets. However, supplementation of tryptophan decreases in fruit fly lifespan in adulthood, no effect, maybe a slight increase in development. And it does seem like genetic manipulation of tryptophan might increase lifespan in fruit flies with no obvious decrease in fitness, but again, very preliminary. I would not publish any of this just yet, but it's kind of leaning that way. So that's kind of one story on how we can use metabolomics kind of across species to get a general novel hypotheses and then begin to test it in the laboratory. And I just want to tell you one more short story. Hold on in time. Yeah, I think we're good. Doing comparative metabolomics of different lifespan extending interventions. So in the aging field, lifespan extending interventions are kind of anything you can do to an organism to make it live longer. These can be genetic, pharmacological, a drug, or dietary, physical exercise. So any manipulation you make to an organism that makes it live longer and has been repeated over and over, and it definitely makes it live longer, is a lifespan extending intervention. And so we've been doing some metabolomics on different interventions, and the question is, do different interventions actually act on the same metabolic pathway? And so I'm going to show you some data that's going to point towards the role of carnitine metabolism maybe in some of these interventions. So this started with a study where we were looking at the effects of temperature on longevity. So temperature is arguably the oldest environmental manipulation that's been found to increase lifespan. So this is a paper from 104 years ago, where they showed that fruit flies raised at colder temperatures lived longer. So as temperature decreased, longevity increased. And since then, this has been shown over and over and over again in many, many species. This is, again, some data in fruit flies, where you're looking at 18 degrees, 21, 20, sorry, 21, 27, 30. So you increase you increase temperature, you decrease lifespan. In invertebrates, this is 100% shown. It's been shown in fish, so in other vertebrates, and then it's also, there's some evidence in mammals that lower core body temperature might lead to increased lifespan, but I won't say that that's been 100% shown. But at least in ectothermic type animals, it seems to be decrease the temperature, increase the longevity. However, why this is actually has not been investigated that much. It's just one of those things that's known. There's been ideas about metabolism and oxidative stress, but no one's really investigated what leads to this. And so what we did was just a small pilot experiment to do metabolomics on flies raised at 3 different temperatures, to ask what is different in the metabolome between these different flies. Is there something that is seen in the lower temperature, longer lived flies that's different than the high temperature short flies? So we ended up looking at about 6,000 different metabolites. And so these are our survival curves. They're not as pretty as this study, I would say, but you still can see significant increases in lifespan as you move to lower temperatures, where you have flies living to 150 days at 18. So then we sampled them at 45 days for the 24 and the 18. So this is right where these two curves are really beginning to split. This is what we were really interested in. What's the difference between the 24 and the 18? The 28s, we unfortunately had to sample at 35 days because at 45 days, they're all dead. So you can't sample them. But we're really interested in this kind of point where these two curves are shifting. So what we found after all this metabolomics analysis, one of the pathways that jumped out are the carnitines. We found that different carnitines appear to vary with temperature. So for most of them, some of them you're seeing lower levels at the higher temperatures, suggesting that longer lived flies have higher levels. Some are showing slight opposites. Again, we're seeing higher at the lower temperatures. However, you also notice there's a huge sex difference in these, which might help explain some of the sex. Oh, does it go the wrong way? Sorry. Some of the sex differences that we're seeing in the longevity in the flies too. So what are carnitines? So these are actually metabolites that help shuttle fatty acids into the mitochondria. So what you're looking at here is, this is the cytosol of a cell. This is the outer mitochondrial membrane, the inner mitochondrial membrane. And carnitines help move fatty acids from the cytosol into the inner mitochondrial membrane, into the matrix, so that it can be used for fatty acid oxidation. So in theory, carnitines help your body use fats for energy instead of carbohydrates. So there might be an idea that an increase in carnitines might lead to an increase in fatty acid oxidation, so you're better able to utilize fats for your energy source. So this was interesting when this came up, and the real reason this was interesting to me at first was that buried in the supplement of my very first metabolomics paper is this figure. So in my very first metabolomics study when I was in grad school, we just looked at what changes with age in flies. And what we found is that carnitines decline with age. And so anything here in blue is something that goes down with age. So we didn't really focus on this in that paper, but it was interesting that there actually seems to be this huge change in carnitines with age, and we're seeing also a change in carnitines with temperature. So maybe there is something related to aging and longevity that might be affecting how your body can utilize fats as an energy source. So then the question is, okay, this is fruit flies only. What about other things? What about other lifespan extending interventions? So I did a small pilot study with Daniel Smith at UAB, and Daniel is really interested in acarbose. And so those of you who don't know about acarbose, it's considered a potential calorie restriction mimetic. Giving it to mice makes males live longer. Females, it doesn't really have an effect, though it does increase health in both sexes. This is a drug that's very well tolerated. It's already given to people as a diabetes drug. So it was an interesting intervention that seems to affect health and longevity. It's one where we really don't know the action through which it's working. And so we did a small metabolomics study on mice that were raised with or without acarbose. And so then we asked what was different. I'm not going to go into the whole study, but we did find that carnitines in general are higher in acarbose supplemented mice. So here are the control mice, and here are the acarbose mice. And you can see that the control mice have significantly lower levels of these carnitines, again suggesting that maybe mice with acarbose can upregulate carnitines such that they can better utilize fats as an energy source, which would also make sense as this is potentially a drug that works on diabetic patients. And so they would need to not use their carbohydrates as an energy source as much. It's also interesting to note that the variation in the control is much, much higher than the acarbose, with the exception of this one little outlier. So again, we're seeing carnitines might play a role in these long lifespan extending interventions. And so what is going on with these in maybe just naturally long-lived animals? So Heather Patterson was a postdoc in our lab, and she's now an instructor who still works out of our lab, is a marine biologist who got interested in aging and came to work in the lab. And she and I did a clam metabolomics study, which might sound kind of weird to some of you, but clams actually show enormous variation in longevity. So the longest lived animal known at the moment is Arctica atlantica, or the mahogany clam. It lives about 500 years, or it can live up to 500 years. These are not huge clams, which is what people think. These are small clams. They fit in your hand. We can raise them up in the lab. These three species that we looked at with their longevities, these are all species of clam that you eat when you go to a seafood place. So these are all general clams. You can find them easily. We actually order them from fishermen who ship them live to us, and then we keep them in the lab to raise them. And so there's such variation in these clams. Heather was interested in understanding what is going on with their metabolome and their metabolic pathways. And so she and I together did this study. And so I just asked her, okay, what's going on with the carnitines, just to see how it might fit within this study. And it actually kind of goes against what we think might be going on. So Arctica, which are the longest lived animals, actually very, very reduced carnitines compared to the two shorter lived species. So this doesn't quite fit our pattern, but these are, of course, very significantly different. So it does at least seem like variation in how organisms deal with fatty acid metabolism might be leading to some of the variation we see in lifespan. But it might be somewhat species specific, environment specific. There's still a lot that we need to kind of understand, again, to understand if this even really matters, or is it just a byproduct of these other things? So we're seeing this variation. So overall, with the carnitines, we begin to see that comparative studies of these different interventions can provide us novel hypotheses on aging and longevity. And they might help us develop hypotheses that we wouldn't have got if we'd only looked at one. Because sometimes when we look at one thing, we tend to throw a lot of it to the side. But if we can look across a spectrum and we see the same thing pop up over and over and over again, it might begin to give us more information to actually go after those pathways. So carnitines naturally decline with age. They seem to potentially be higher with some variation in response to interventions that extend lifespan, but they're also very low in the longest lived animals. So there's still a bunch that needs to be teased apart to understand what these different metabolites might be doing. But I think we have enough information now to kind of go at it and see what's going on. So in the future, we're interested in how tryptophan actually changes in response to lifespan extending interventions. And this is a big crux of my K99 that I was hoping to have more information for you. But as you know, we again got shut down. So I've got a lot of mouse tissue from mice that are growth hormone mutants that are sitting in the freezer waiting to have some molecular work done on it. Because what we're hoping to see is similar patterns in growth hormone reduced mice that we see in small dogs. And that might give us some sort of an IGF-1 independent pathway that actually is leading to small size and long life. Because we're constantly seeing this idea that within a species, you reduce body size and it lives longer. And so we're hoping that we can find pathways that are conserved across these species that show this size longevity trade-off. And then we're going to keep working with the flies. These are my students that are doing these projects to really get at the more genetic manipulation effects of the tryptophan metabolism pathway. And this leads to manipulation of it, and can we increase lifespan and health in flies, and what are these fitness consequences, because we know there's got to be some somewhere, so can we find them? And then lastly, we're hoping to look more at the carnitines to see if they have a direct effect on lifespan. We have some very, very preliminary data that's similar to the tryptophan study suggests that supplementing carnitines, it's not negative, but it has no effect. So it seems like supplementing carnitines directly might do nothing, but it's very preliminary. And so we're going to follow up on that. It's something I was hoping to get to more this year, but because of COVID, I don't have nearly as many students to do all the different experiments. So as you can imagine, this has been a kind of project that's had lots and lots of different people because we pull in lots of different species. So I had a group working with the dogs and the flies. I want to point out Tyler and Zoe, who are my two undergrads right now. The two of them, all of the new tryptophan stuff, they did all of it. I helped them set it up, but it was all of them. They're great. Daniel Smith helped with the mice. The marmosets were done with a different group. Finally, a last group with clams that was started by Heather Patterson, like I said, in our lab. The dog work was funded by our postdoc fellowship, now into my K99. The rest of it was funded by different NIA sources in the K99. And I always have to put in, this is my dog, Aspen, who is not in my study because she's terrible to get blood from, but I'm hoping to make her maybe live longer in the end. And so with that, I will take any questions on my own time. I guess I'm okay.
That was great. Perfect. There's a lot of chatter in the chat box. A lot of
Okay,
questions.
good. I know I talked too fast, so please, if I didn't explain something, let me know.
No, that was great pace. Howard, you had a lot of questions. You want to ask one of your favorites to start with?
Yes. Like to start off with the dogs, I noticed that there was like a steeper decline between longevity and size in purebloods or purebreds versus mixed breds. Could that indicate a tighter relationship between them, or?
Yes. So it does. Let me go back over here. You're talking about this one?
Yeah, that one. Yeah, that one.
Yeah. So yeah, we haven't published on this, but it does seem like, so it's been published before that purebreds live about 1 year less than mixed breeds. But the data, when you break it down, it really looks like the real effect is in your large dogs. Your small dogs don't really seem to show that big an effect, which I think makes sense. So the longest lived breed on average is the Chihuahua. They live about 16 to 17 years. And a mixed breed at that exact same size is going to live about the same. And I think it's something that we probably need to really analyze carefully and potentially write it up, because I do think this is real, that the mixed breed matters a lot more in the large size than it does in a small size.
Yeah, I guess as I understand, purebreds are more inbred than mixed breeds. So it's kind of teasing apart like the size versus the genetic variation.
Yeah. And I think it's going to be a hard thing to do, but I think it needs to be done. And I think it's really interesting that we're seeing this pattern. I, of course, didn't put all the dots on here because then it would just be a cluster mess. But yeah, and I don't know. I don't know what it is genetically that might be leading to larger purebreds dying earlier, especially because some of the large breeds are actually some of the least inbred. So the labs and the Goldens are some of the least inbred breeds, but they're big and they die fairly young. So
Well, they could also be like more stress on their bodies.
it could be, yeah, it could be something like that. I think there's a lot of potential answers, and I don't know the answer, but I think it's really interesting.
Yeah, we
Cool.
got time.
Thanks.
No
Do
problem.
you want to ask one more, Howard?
Sure. Sure. I was kind of curious about that relationship between cold temperature and the carnitine teens. Like at cold temperatures, are fats a better energy source than carbs? Are they more stable, or is it easier to metabolize those, or is there something else going on? Like kind of how far back does this go?
Yeah, I don't know for sure. And I wouldn't call these cold temperatures. Like 18 degrees is not that cold, right? So it's not like some of the studies that have done metabolomics at really cold temperatures, like 6 to 8 degrees, and they see very different things. So that's a real stressful environment. 18 is not that stressful for the flies, but I'm thinking it maybe has something to do with the utilization of fats is just a better use for your own health, is what I'm thinking. And the low temperature flies are better able to do that, and that may be indicating why they're leading to a longer lifespan. One thing I think we need data on, which I don't know how I'm going to get it, is like mice that have been raised on like a ketogenic diet or a high fat diet, to see what they have going on. And I think that will really kind of maybe help get at what is, and I could do flies, like we can put flies on a high fat diet, but they don't do well, actually. They don't like it. But I think that's just a physiology of the animal. I think mice, though, on a ketogenic diet would be a really interesting question that I think would really help answer some of what's going on. And like I said, it could just be everything as a byproduct and means absolutely nothing. That's one of the issues, I think, with these large omics studies, is that pinpointing what actually matters is hard.
That's absolutely right. I would add that 18 Celsius might actually be very cold for an Arizona fly.
That's true. I mean,
Not true.
and they do move less than like a 24.
Right, certainly.
Fly,
It's better than high heat.
Yeah.
So Laura Jameson asked the question. Laura, do you want to ask it, or I can read it out loud? I'll go ahead and read it. Are the female Drosophila more sensitive to tryptophan, and do you have any theories why? So in your tryptophan experiment, where you had it?
Yeah, it looks like the answer is yes, but I have no idea why. Yeah, it's something we haven't thought about that much yet because it's new, but it's something we're interested in because we're very interested in sex differences in our lab. It's interesting to me because in general, in most genetic backgrounds, the females are already longer lived than the males. So I don't know why they would be more sensitive to it, but it's a good question. And once I can get some students to really get at some of this, I think it's something we really need to look at. And I really just don't know why. I don't know enough about, there's not enough in the literature about real sex differences in tryptophan metabolism yet.
Cool. I have a question. There are some more questions by students, but we can get to those in the next 30 minutes. My question is also about the sex differences, because you saw in your temperature study, you saw really large, you saw differences across temperatures, but you also saw sex differences within temperatures. Are those sex differences in the same direction as you would expect with respect to like the sex differences in lifespan and then the temperature differences in lifespan?
Sorry, can you say that again? The dog barking in the ear.
Are the sex differences in, so females live longer than males, right? And yeah. Animals at colder temperatures live longer, right? So do you see that in metabolites where they have lower levels at colder temperatures, do they also have lower levels in females?
So I haven't looked specifically at that exact pattern. We do in our model run the sex by temperature interaction because that does, and there's a lot of those, but I haven't gone back to see if the patterns correlate with each other the way you would expect.
Right.
And I think that's something, that's a great idea that I'm going to write down right now.
It just made me think, like, if we think about something recapitulating aging, or if we think about differences in like certain effects having, certain variables having similar effects on a phenotype like aging or longevity, then you hope that their effects are concordant, or might be different mechanisms that are leading to sex differences in longevity that are leading to temperature differences in longevity.
Yeah, and part of the reason we were interested in the sex differences is, in general, females are longer lived, but should have done it the other way. You can see we actually get a switch, and we get this, I didn't talk about it, but it's something we've been looking at. The females are longer lived at your standard temperature and at the warm, but you actually get a switch. And we see this over and over where it looks like the males are actually longer lived potentially at the colder temperatures. And so that's something we're also trying to tease out what's going on. It's just, it's so much data, and it's also like what matters, right? Again, most probably
location,
doesn't.
right?
Yeah.
Cool. So thank you, Jessica. Does anyone who will not be sticking around for the next 30 minutes have a question they would like to ask Dr. Hoffman? If not, I hope everyone will join me in thanking Jessica for a wonderful presentation.
Well, thanks for having me.